I had a really good experience in the SIT program in 2008. I actually stayed about 3.5 months, so longer than most of the students. In fact, we finally just published the results from the study that I conducted during that summer. And even while I was still in Taiwan during the SIT 2008 program, I already submit my postdoc application to come back to Taiwan. So the timing was perfect. I basically left Taiwan in September, submit my application to the National Science Foundation, and then a few months later they told me that they would fund me to come back to Taiwan for this for 2010. So the timing worked out perfectly for me. And now I'm right back where I was when I did the SIT program. We study, and when I say we, it's me and about 6 or 7 other professors. We really are interested in kind of the basic biology of reef corals. So the corals, the main animal that makes the coral reefs that most of us are familiar with, is actually a plant and an animal mixed together. It's not, so a lot of the techniques and approaches people have typically used in the past, they just focus on the animal or they just focus on the plant. They don't really consider the fact that it's both. So we try to develop technologies and techniques so people can actually study the endosymbiosis. And endosymbiosis is basically when you have one cell, like the algae, living inside of another cell. So we basically use molecular and cellular biology tools to try to understand the physiology of these corals at the cell level. The main reason for this is there's a big push now for us to better understand how corals will respond to global climate change, particularly the increased temperatures and acidity that they'll face in the future. But in order for us to understand how their physiology might change, we need to have better tools to understand it. So now, kind of at the same time, we're doing manipulative experiments to see how the corals will change their physiology in response to higher temperatures and higher CO2, while at the same time developing tools and techniques for studying their physiological behavior. Well, molecular biology, it moves at a snail's pace. You basically need to do the same thing over and over again and have luck on your side. But slowly but surely, we're getting some pretty interesting results. We're actually doing some large gene sequencing projects right now that I'm excited about. They're expensive, but I think the amount of data that we're going to get back is going to really help push the field forward. It's going to be advantageous to all scientists who want to work with coral. So we kind of see it, it's almost like doing a service for the field. We're kind of doing the work that nobody wants to do because it takes so much time and money, but in the end it will help, it's going to help everybody get a better understanding of these reef corals. So you just need to have a lot of patience, basically. The research environment actually at this institute is great because it's probably one of 2 or 3 places in the world where you actually have the marine life, all different kinds of corals, fish, marine creatures in this building, just 50 or 100 meters away from where we're doing this interview. You've got all different means of manipulating their environments, and then you can bring the samples just back to this building and do any kind of sophisticated molecular analysis. So there's only a few places in the world that have that convenient of a setup to do this kind of research. In terms of the science kind of system in Taiwan, it is a little bit more top-down than I'm used to. And what I mean by that is I feel a lot of the students, unfortunately, I feel like they don't really get to develop their projects as much, whereas in America, if you can develop your own project to kind of go in your own way. But here I feel like the professors have a lot more control over the projects, and I don't think it's necessarily better or worse, it's just a different system. But I think the main reason for that is there's much more master students. I think once the aquarium gets more PhD students, they'll have more training and they'll be able to kind of develop their own projects. But right now it's mostly master students and they're still kind of at the junior level, so they don't really get to have a lot of their own ideas. They're just kind of the workforce. So I hope that will change in the future when there's more senior students. I would say one thing that I really like about the aquarium is that they have a very exploratory approach to science. And what I mean by that is we're always trained in America that we need to have, kind of a hypothesis. We need to have a question with a specific idea in mind, and then we go down a path to test it, which is fine. That's what science is about. But there's another part of science where you just take an animal, a coral for instance, and you want to see what's there. What is it made out of? What are the different kinds of cells? You're not actually asking a research question, you're just exploring something about the animal. And they do a lot of that here, where they're characterizing, for instance, unique structures of coral. And they're not actually, doing an experiment like changing the temperature or changing the oxygen environment. They're just looking in the tissues and cells to see what kind of interesting things they can find. And in America, if you try to do that kind of research, they may not respect your science as much because they don't understand, you don't have a hypothesis. But actually I like the fact that here you can do this kind of exploratory science and then also the hypothesis-driven science. Whereas in the US, you need to have a hypothesis. Very rarely does the National Science Foundation fund kind of exploratory projects where you just try to search for something and you don't really know what you'll find. So it's different, but I think both systems can work. One thing I really like about the environmental politics in Taiwan is in America you could have the best science in the world and it may not make its way into the common people or the government. Nobody may know about your research. But a lot of the aquarium researchers I've found have been really impressed by the fact that they find some interesting result that they think could help save a coral reef, for instance. They take this information to the national park and then they take it all the way up to the highest levels of the government and they say, look, this new hotel, for instance, you're going to build here, it could destroy the coral reef. You need to stop. And changes can happen. This kind of change where you take the science directly and try to make some kind of management decision, that's the goal for a lot of countries, but it never happens. And Taiwan is one of the first places I've seen where the scientists, they take their data, they show it to somebody in a government official, and they say, this is not acceptable what you're doing. So for me, working on coral reefs, which are especially sensitive, I've really been impressed by the fact that people are doing science and then making sure that the public and the government officials know about their data. And I think this is something about the Taiwanese system, at least in my experience, that I really like, because I wish it could work like that. I will work here for at least another year. I really hope that in the meantime I can find some other funding source that could potentially keep me here longer. So it just kind of depends on the financial situation. But I've spent probably 12 months of my life in Taiwan at this point, and I still see it as a place where I like to stay. So I hope in the future I can, if I don't get to stay here, I can at least come back occasionally to do projects, like maybe just a collaboration.